Good morning. Sorry I couldn't be there with you today, but I'm still very glad to be able to submit this little video presentation talking about our research interest in harnessing the soil microbiome for increased economics and resilience. So I wanted to start out with something I think about a lot, which is how can this relatively new frontier of trying to understand the microbiome, the microbial communities in the world around us transform agriculture. And really I see two fundamental ways. The first is in data, so analytics. So what information can we actually extract from the microbes that are out there, specifically in our field soils, that can actually be used for decision-making by farmers? I envision a world in the future where we could get all sorts of microbiome type reports that could guide fertilizer application levels or service soil health indices. But those are all need a lot of research and they're a little ways off yet. But we have a few ways we're interested in this area that I think could be implemented now that I'll talk about in the first half of this talk. The other probably more exciting way is, could we actually improve productivity using the microbiome? So this has been something that's been of interest for a long time. And really it breaks down into three fundamental ways we might do this. One is through management, so understanding a little bit more how management affects microbial communities and how those feed back into productivity. Another is through the host, so can we actually understand how the host controls its microbiome, and then perhaps even breed improved cultivars that have elite abilities to recruit beneficial microbes. And the third is through directly intervening with microorganisms themselves, which is probably the one that's been applied the most in the past, in some cases, such as with rhizobia inoculants to great success. And in others, a little bit more like snake oil. But we're really interested in investigating all these areas, especially the host and the microbe in my lab, and trying to figure out how can we actually deploy the microbiome to maximize productivity within cropping systems. So I want to start the talk today talking about informatics to guide grower decisions and our interest in that area. And that interest specifically relates to rhizobia and the inoculation with bacteria that actually can take nitrogen directly from the atmosphere and supply it to soybeans. So I think everybody's probably aware of the crazy prices that nitrogen has gone through in the last year. I think those economic shifts and the costs of fertilizer really are pushing us to make sure we're maximizing symbiosis with rhizobia, which can supply most of the nitrogen that soybean needs. These are sustainable alternatives to nitrogen fertilizer. However, inoculation isn't always required. So a lot of research, especially some great research at Carrington REC, is suggesting that after you've established rhizobium populations in your soil, at least for soybeans, these can continue to provide the benefits for many years afterwards. That being said, there's a lot of different factors to think about when it comes to how long, are you safe at those populations you have, or do you need to start re-inoculating? So these include, for example, the number of years since previous inoculation. So we know 5 years you can get away with it. How many more, we're not really sure. But other factors like flooding or say drought like we had last year, those could certainly cause crashes in your population and maybe cause a need to re-inoculate, or things like non-optimal pH, or we've been thinking a lot about as well soil conditions just across the state and how those might influence these sort of general knowledge about inoculation. So we've set out to develop a fast and cheap molecular assay for rhizobia that could just very quickly, very cheaply quantify how many rhizobia were in a farmer's soil and just give feedback in terms of whether we would recommend inoculating that field or not. So you can think of this like an auxiliary to the soil test, giving you biological informatics about those beneficial microbe populations. So we've been developing an assay using quantitative PCR approaches that actually target symbiotic genes in the rhizobia and can tell us how many of those rhizobia are there in the soil. We're working at optimizing the sensitivity and specificity of these assays as well as sampling methodology at the moment, but we are making some progress. So just to show you some preliminary data, what I'm showing on this slide on the left is just a simple greenhouse assay inoculating with different amounts of rhizobia from 0.1 rhizobia per gram to 1 million, and looking at the response of the soybean in terms of how many nodules it produces, and kind of trying to find a threshold where we get a maximal symbiosis, which we see in the greenhouse around 1,000 rhizobia per gram. Then on the right-hand side is our molecular tool quantifying the number of rhizobia per gram on the y-axis based on a signal that comes from a qPCR machine on the x-axis. And you can see a standard curve we've established there that works very well, both with genomic DNA, which we started with when we just kind of dilute that out to different concentrations, but also when we spike soil that's naive, doesn't have rhizobia in it, with known amounts of rhizobia, we're able to fairly accurately predict how many rhizobia are there. So when we then take a field soil sample, we can just apply that onto the standard curve and determine how many rhizobia per gram are in that field, and hopefully then make recommendations regarding inoculation. We're working now to improve the sensitivity of this assay. We can detect down towards between 1,000 and 100 rhizobia per gram. We'd like to get that closer to 10 or 1. Nevertheless, I think this is, it's going very well. And one of the things I wanted to highlight with this is we'd really like to start sampling farmers' fields this year. So if you're interested, please get in touch. I'll leave my email at the end of the talk. I'd love to come out and sample farmers that are interested in their fields and determine how many rhizobia are there. So that's just my brief window into informatics. Now I'm going to go into sort of the meat of this talk, which is talking about harnessing the microbiome for plant benefit. So probably many of you are aware that plants have their own microbiome, just like we do. That's broken up into a number of different compartments. We have the phyllosphere, which is in the above ground tissue of the plant. The endosphere is inside the roots of the plant. The rhizosphere is in the soil adjacent to the roots of the plant, and that's where a lot of the action is in the plant microbiome. And then, of course, we have the soil outside of that. Interestingly, the figure here on the right side just kind of highlights the genetic potential of the microbiome compared to the plant itself. So, compared to a tomato genome, which might have 42,000 genes, the bacterial symbiont genome of the tomato could contain as many as 100,000 or 1 million different genes that could all be at play and harnessed for improved productivity, potentially. So we've become very interested in iron deficiency chlorosis. I know Goos last year gave a great talk on this. He worked, did an amazing job pushing the understanding of IDC in North Dakota throughout his career. We've benefited a lot from that understanding, and we've basically become focused on this as perhaps this is an area where the microbiome could intervene for improvements. So IDC is caused by high levels of lime in the soil. It's a big problem in North Dakota. It's made worse by topology of the soil and nitrate, salinity, high pHs. And it's not necessarily a lack of iron that's present, it's a lack of iron that's available to the plant. And this is certainly an important stressor of crops because it causes $260 million or so of yield loss annually in the northern US. So there are different ways to combat IDC. I know Carrie Miranda is working very hard on breeding, improving crop genetics. And certainly this is an avenue that will likely show some gains, and it's very important, but I would suggest that perhaps it's not a complete solution. So there's still many varieties that have great other traits that aren't necessarily completely IDC tolerant. And on this graph here, it's just showing a number of different varieties and their IDC ratings. We generally see a 9 to 19 bushel per acre loss in yield per chlorosis unit when we're quantifying iron deficiency chlorosis. So these numbers on the left, 4, 3, 2, 1, those are indicating how chlorotic are the plants. And you can kind of do the math there to see how much yield loss you're getting. So iron chelating fertilizers are an option. So one called Soy Green, for example, does a great job. They are chemicals that in the soil can help alleviate bound iron and make it available to the plant. This can be applied, for example, at 3 pounds per acre in furrow during planting, but it's expensive. So $18 an acre is not an insignificant input. And certainly if we could get to the point of microbiome replacing these fertilizers, I would expect costs to be much, much cheaper for a microbial-based product. Or, it would be nothing if it could be plant genetics that recruits a beneficial microbiome, for example. So the microbiome has a lot of potential in this way. One of the reasons is because bacteria are very good at freeing up bound iron and making them available. They use chemicals called siderophores. And if you have a very keen eye, you'll see the structural similarity between these siderophores and the soy green chemical. They use similar groups to free up bound iron and make it available. And this can definitely be something that could be at play for making iron available to the plant. We also know that, or we're starting to understand that plants are able to recruit beneficial microbiomes when they're needed. So they do this in a strategy that's being called a cry for help strategy. And really this works by a stressor and a plant detecting that stress and responding to that stress by secreting chemicals generally that can alter the composition of their microbiomes. So in response to abiotic stress, the plant can send out this cry for help, usually perhaps a chemical molecule in their root exudates that go out into the soil and can actually shape the composition of their rhizosphere microbiomes, for example. And so this is an avenue that we're starting to learn. It's a tool that plants have to respond to different stresses in their environment, be that things like drought or iron deficiency, for example. And so there's all sorts of biotic and abiotic stresses that have been investigated. This is an overwhelming slide, but just kind of summarizes a lot of the really interesting research that's out there, looking at different stresses and different compounds that the plants produce in response to those stresses, and the ways that different microbes respond when the microbiome is being restructured by the plant. So I'm going to just zoom in on iron deficiency here. There was a really great paper in 2020 in Cell Host and Microbe looking at the model plant of Arabidopsis and these molecules called coumarins. So they found that in response to iron deficiency, Arabidopsis biosynthesizes these coumarin molecules. It releases them out into the rhizosphere. And the coumarins themselves play roles in mobilizing iron. So they help to release that bound iron, but they don't do that job alone. Once they've released that bound iron, microbes actually take it kind of a level further and make that available to the plant for import and then use under iron deficient conditions. And so they found a link between the coumarins that were excreted under iron deficiency stress and the restructuring of the microbiome to have more microbes that do this job of mobilizing the iron for the plant. So there is emerging literature out there showing us that plants have the potential to restructure their microbiomes in response to iron deficiency in a beneficial way that can actually help alleviate these stresses. And so this is really exciting and kind of fueling our interest in this area for the microbiome. So we have started a study on this, collaborating heavily with Carrie Miranda, who's been generously planting field trials for us. And this is just sort of our preliminary study that we harvested last field season. So we have 4 different varieties, 2 that are sensitive to IDC and 2 that are tolerant, really just to see if we see differences depending on those phenotypes of the plants. And we've grown those in 4 locations. 3 are locations with IDC and 1 is a location without IDC. And we're basically then having a look at the microbial communities there, assessing their composition, and we're trying to understand, do we see evidence that the plant is able to restructure its microbiome composition during iron deficiency chlorosis? So do we see different microbiomes in the IDC minus condition versus the IDC plus conditions? So this is just on the right, a picture of Carrie when we had a nice trip out there to have a look at the plots that they'd planted for us and flagged them out for sampling. On the bottom, you can see some of the different varieties that in the IDC conditions at Leonard. And the graph is just showing the IDC ratings that we gave to these plants that were in these different locations. So you can see that we saw a nice IDC response in the locations that were IDC plus, especially Leonard, which had the most pronounced IDC, but as well Castleton and Colfax, particularly in the IDC sensitive varieties, the ND167108 and the ND17009 GT. And then in Prosper, which is the IDC minus location, all the plants did quite well and didn't really have IDC at all. So great, we have a really nice set here of samples that we could start to investigate this question as it's occurring in the field. And so actually this is data that we've only just got back yesterday. So we've spent a lot of time optimizing our microbiome pipeline in the lab, but finally it's up and running and working really well. So this is our preliminary data for these samples. And what I'm showing you is a graph that basically shows you the relationship of the microbiomes in each of the different samples compared to one another. So the closer those points are on the graph, the more similar those microbiomes are to one another. So if you just kind of know that and visualize this, and I tell you that they're colored by location, the color and the symbols represent the different locations, you can see that microbiomes from the soybean rhizosphere, so from the soil on the soybean roots, really cluster very nicely according to where they were isolated from. Now the axes on this are associated with variables that influence that composition. So the x-axis is one unknown variable that's influencing the composition of the microbiome. And the further apart they are on that x-axis, the more that variable has influenced that particular sample. And on the y-axis is the second variable that's showing the second most influence on the microbiome. And what's really exciting to me is if you look across the x-axis, so the relationship of the samples from left to right, we actually see a segmenting of the sample according to the IDC level in the field. So the least IDC field is the furthest on the left, and the most IDC field is the furthest on the right. So this is pretty exciting preliminary data because we're maybe seeing those IDC effects in the soybean rhizosphere, and we may be seeing different microbiomes there in the rhizosphere of soybeans when they're grown under IDC conditions compared to when they're not. When we look at the data by variety, so now this is that same graph, but now the colors are by variety and the symbols are by location. You can see we don't see a lot of difference whether the variety itself is IDC sensitive or IDC resistant. So they may, if they are restructuring their microbiomes in response to IDC, they may be able to do this equivalently, regardless of if they're a sensitive or a resistant variety. And one of the things then that we need to start thinking about is, can we provide evidence that this effect really is caused by IDC, that these differences in the microbiome are really caused by IDC? And we'll start to do that as we start to look at comparing the rhizosphere microbiomes to the soil microbiomes. But ultimately, it's pretty challenging when you have different soil types to conclusively prove that the restructuring might be in response to IDC and not simply just in response to different starter inoculums from the different fields. So to dive deeper in that, we've actually been carrying out a greenhouse assay. Goos was actually incredibly generous in teaching us this greenhouse assay for IDC that he's developed across his career, and I view as an incredibly important contribution to the field going forward, and we're certainly going to benefit greatly from it. So we've successfully executed this assay based on what Jay told us. And when we did this assay, our objective was to actually design an experiment where we could actually divorce the soil type from IDC. And so the way we did this was, instead of using different soils that had different IDC levels, we used a single soil. So we used the Leonard soil that has the high IDC. And then we used that product soy green to actually alleviate the IDC from some of the pots. And hopefully, when we sequence the microbiomes of these samples, this will give us a way to visualize specifically restructuring of the microbiome that's happening from the same starter soil when the soybeans are under that IDC stress versus when they're not. So this is a really exciting experiment we've carried out now, and we're ready to start sequencing those samples, and I'm really excited to see the results there. So just to wrap up, we are working on developing tools that can allow us to give informatics to the farmers based on the microbiome. Right now, we're just focusing on groups of beneficial microbes, specifically rhizobia. And we'd like to see these tools really actually able to directly guide farmer decision making. And even if not, they should accelerate agronomy research for us, so we can much more quickly start to test whether different treatments affect rhizobium populations. So how do crop rotations influence the rhizobia and how stable they stay in, or how does intercropping, for example, affect that? Some of these different questions that farmers might be interested in. The second conclusion is that we are seeing intriguing differences in the composition of the soybean microbiome across this transect of iron deficiency chlorosis in fields. We're working to dive deeper into that to provide conclusive evidence about that by employing a greenhouse assay, for example. And then the next big question is, can we actually harness this? So we will certainly be transitioning to isolating microbes that we see are enriched and evaluating whether they might actually have potential, maybe as inoculants, or maybe through breeding implant genes that might be involved in recruiting them to actually alleviate IDC using this sort of new avenue to combat IDC, the microbiome. So I just want to finish by acknowledging the contributors to the work. So within our lab, Urmi Das is a PhD student who did most of the effort, most of the work that I talked about today, and Megan Ramsett, our research specialist, also contributed a lot to this work. I also want to acknowledge our collaborators, Carrie Miranda and her team for planting those trials for us, hopefully again this year. And Jay Goos for passing on his wealth of knowledge about IDC and these, this phenomenal assay that we can use as a tool in our research now. And finally, I certainly couldn't have done this without funding, and the North Dakota Soybean Council has been very generous in supporting both of the projects I talked about today, so we really appreciate that support. So thank you so much for your attention. I'll just leave by saying, if you have any questions, since I'm not there to answer them, unless I happen to be sneak poking around in the chat right now, feel free to email me as well if you're interested in collaborations, or also anyone that might be interested in having their field sampled for rhizobia population. We'd be very thankful for collaborators out there that we could come and sample your field this spring. So please get a hold of me if you have any interest in any of those. And finally, I'll close by saying, sorry I can't be there. So I'm probably right in the process of bringing my new daughter home from, Maeve home from the NICU right now. So I have a good excuse. Hopefully I'll be seeing you all next year. But thanks for your attention anyhow.